Hi, hello. Uh, I'm still waiting for people to join. I think people are taking their time. Um, that is okay. I will wait to see if someone is there. And I did check the questions that have been sent. There are not many questions, so I'll wait for people to join in and give us the questions. Maybe anyone is joining. So I was thinking maybe we may have to uh, review contents of week 9 and 10. So essentially cerebellum partly and so still wait for still check if people are joining. Um, Usually we have some good response. I think maybe people are thinking that the class is at a different time, maybe. Not sure what's going on. So we'll be focusing on cerebellum and BG and about 5-6 classes of basic ganglia. Alright. So, okay. Alright, so let's review this content on cerebellum as people are joining in. When people join in, they can ask questions. I'll try to review the contents of the last 2 weeks in the time that is available to me. As people are joining in, they can actually go through, ask questions, and then maybe we can take the discussion from that point onwards. Just remember what we already know. We have already seen probably cerebrocerebellum and spinal cerebellum maybe. And now we'll be looking at vestibular nuclei and flocculus. So essentially, flocculonodular lobe is also called as uh, the vestibulocerebellum. So this is this loop. That part of the cerebellum is called the flocculonodular lobe, also called as the vestibulocerebellum. So in this case, so this part of the cerebellum takes inputs from the vestibular nucleus and actually outputs to the vestibular nucleus. This is very special about the vestibular cerebellum in the sense that the vestibular nucleus is both its input nucleus as well as its output nucleus. Of course, not the same regions within the vestibular nucleus acts as the input and output. It turns out that different regions of the vestibular nucleus act either as the input or as the output, depending on the situation. But the equivalent of the deep cerebellar nucleus, the equivalent of the deep cerebellar nucleus, um, for the vestibulocerebellum is the vestibular nucleus itself. Okay, so in other words, like you have the dentate nucleus, like you have the dentate fastigial and interposed nuclei, which act as the deep cerebellar nuclei. These are embedded sets of groups of, tightly uh, grouped neurons that are deeply embedded within the cerebellar cortex, not the cerebral cortex. Note the difference. That are deeply embedded within the cerebellar cortex. Such a deep cerebellar nucleus is not there for the vestibular cerebellum. Instead, you have the vestibular nucleus, which acts both as the input as well as output. This is very special. This is a topic that I like, and I actually don't know what's on the exam, but I usually test topics that I like. So this is a topic that I like. You might want to make sure that you know the nuances of the cerebellar anatomy in this case. Something that I love to test the students. And also, we had focused on eye movements that we did some time ago, and specifically this case of vestibular ocular reflex. When what happens during in the presence of light and in the absence of light, and how normal fixation happens only during rightward rotation when leftward vestibular ocular reflex is suppressed, but not during leftward rotation. So there are going to be jumps in the movement, okay, fixations. So important to note is that the vestibular cerebellum is found even in fishes. It is the earliest cerebellum. Okay, so it is the earliest form of cerebellum. Note that when something is early, we usually call it as paleo. But in this case, the paleocerebellum is the spinocerebellum. So the earliest cerebellum in this case, we call it as the archicerebellum. Archi, very very old. Archaeology, as in archaeology. So this archi in this word actually shares the root with words such as archaeology, something that must be really old, extremely old. Okay, so phylogenetically speaking, this early cerebellum, so this vestibular cerebellum is quite old as a structure. It's found even in fishes. Okay, so that means it must have some very primitive functions such as balance and so on and so forth. Right? Obviously, vestibular system being vestibular system, it is going to send some very high quality information about the nature of the orientation of the head. So this is one of the early structures of the cerebellum. Then you have spinocerebellum, which is the paleocerebellum. Paleo, although refers to old, in this case is relatively newer when compared with the archicerebellum. So vestibular cerebellum is the oldest cerebellum phylogenetically. Then you have the spinocerebellum, which is found in mammals. This, but then it's only the advanced mammals that have the neocerebellum or the cerebrocerebellum or the pontocerebellum. Okay, so only the advanced mammals such as primates and others, maybe other advanced mammals too. So you have this nice evolution of cerebellum from the early to the relatively newer forms. So what we have noted is that you have the early cerebellum, and this is going to help in balance and eye movements and vestibular ocular reflexes, and this is the archicerebellum. Something to note again, these are the kind of things that might be asked in exam. Someone is asking, Manmohan saying, "Hi, what is this?" Manmohan, I just started reviewing, content on cerebellum in the last 2 weeks, weeks 9 and 10 classes, because there was no questions. There was nobody who had joined. I thought I'll start reviewing some content as we go along. If there are some questions, I'll stop and answer them. But if not, I think I will proceed. Manmohan, do you have any questions? Okay. Note that in note that there are direct and indirect pathways through which information from the spinal cord reaches the cerebellum, and how spinal cerebellum is going to help modulate movement. And the key word here is modulation. And so, why? Because cerebellum is that part of the brain that is more involved in modulation, coordination, modification of movements rather than control. Technically, control means there is a very tight coupling and there is a one-to-one response between the input and the output. Technically, whereas in the cerebellum you are going to have modulation or coordination rather than one-to-one response. So information reaching from cranial nerves 3, 4 and 6, what are these? Please check what these are. And information coming in from vestibular labyrinth and sent to the flocculonodulus lobe, and which then reaches the vestibular nucleus, which is the equivalent of the deep cerebellar nucleus for our case, which then sends information back to the spinal cord via the vestibulospinal tract. Note that this is a unique case of when a deep cerebellar nucleus, if the vestibular nucleus can be considered as a deep cerebellar nucleus, when the vestibular nucleus directly contacts the spinal cord. This is quite this is quite unusual. Why would this happen? Because, there may be cases when this is needed, such as posture, balance, and retention of balance, and so on and so forth. Okay. Find out what these cranial nerves 3, 4, and 6 are. If you haven't watched the videos, I think you should watch the videos. Okay. Note that information from the spinal cord reaches the spinocerebellum via multiple pathways. One is via the spinocerebellar and the cunocerebellar pathway. The other is information from the spinal cord reaches the inferior olive. From there, you have the olivocerebellar pathway, okay, from which is essentially the climbing fibers. So that's one. But even within the spinocerebellar pathway, you have two pathways: the dorsal pathway and the ventral pathway, and then cuneo-cerebellar tract. Right. So this is how the spinocerebellum receives its inputs. What are its outputs? Its outputs are many actually. One to the ventrolateral thalamus, which then projects to the cerebral cortex, which then projects to the corticospinal tract, which then projects to the spinal cord through the CST, corticospinal tract, also called as a pyramidal tract. Remember, long time ago we saw this as a pyramidal tract, and engages in control of movement. So this is the crucial role of the cerebellum in acting as a modulator of movement. So receiving input and sending the corrective information to the cerebral cortex, and the cerebral cortex will take action. But that's not the only one, because it turns out the cerebellum then projects to the reticular formation. So essentially there are also brainstem nuclei that control movement. These are the reticular formation and red nucleus. And reticular formation projects to the spinal cord via what is called as the reticulospinal tract, and the red nucleus projects to the spinal cord via what is called as the rubrospinal tract. Note that there are in reticular formation, there is also information that is received from the spinal cord from various nuclei such as the reticularis tegmentopontis, paramedian reticular nucleus, and lateral reticular nucleus. So this information is also sent in a two-way street from the spinocerebellum. So reticular formation sends information to the spinocerebellum, also receives information from the spinocerebellum. So from this, what we understand is that the cerebellum essentially receives information from multiple tracts from the spinal cord, but projects to the spinal cord indirectly, only indirectly, either via the vestibular nucleus or via the red nucleus or via the reticular formation or via the cerebral cortex. Okay, so indirect projections, which means so essentially modulation of movements. And there are some organizational principles associated with spinal cerebellar this, and what is the possible role of parallel fibers in movement coordination, right? So there are three principles stated in Kendall and Schwartz that we discussed. One is both Purkinje neurons and deep cerebellar nucleus neurons discharge in relation to voluntary movements. So voluntary movements and DCN neuron activity and Purkinje neuron activity are very well modulated. Okay. Second is that there is an anticipatory control of muscle contractions. So the timing of movements is well controlled. So this is actually a very interesting principle. So timing of movements is probably modulated by the cerebellum. And then third one, relatively controversial one, is that there are internal models of movement dynamics of the limbs that take account of limb structure. So these are the three principles that we have discussed. This is an example of the case when the internal dynamics is taken care of. The internal models take care of the interaction tasks, for example. This is the example when the absence of cerebellar damage, what does it do to proprioception and proprioceptive information and the inability to account for interaction tasks, for example. And note how parallel fibers act. Parallel fibers, how do they project? Let's remember, they have a sheet. So suppose that is the sheet, and suppose that is the, what is this? What is the example that I am trying to give here? This is the Purkinje fiber dendritic tree that is spread out very widely in 2D. But when I look at it in the third dimension like that, all I am having is a very thin layer. So this is the Purkinje cell's third dimension, that's very thin. And the parallel fibers are going through this dendritic tree in a direction that is perpendicular to this. If not, so how many you have? You see that the Purkinje cell's dendritic tree has, anywhere between 250,000 dendrites, or around 250,000 dendrites. 100,000 to 500,000 dendrites, around 250,000 dendrites. So 250,000 dendrites, and these these parallel fibers that we are talking about, right? Remember, the parallel fibers. What are these fibers? These are the axons of the granule cells. Right? Not the granule cells in total constitute more than 50% of the total number of neurons in the entire brain. So only one type of cell in the cerebellum accounts for more than 50% of the neurons in the entire brain. We are talking about billions of these. Then a question is, what could be the purpose of this? You are having billions of these axons that are going perpendicular to the Purkinje cell dendritic tree, and the Purkinje cell has a very intense and a profound impact on the activity of the deep cerebellar nucleus, because it inhibits the activity of the deep cerebellar nucleus. Purkinje cell output is GABAergic, remember. Okay. So it inhibits the activity of the deep cerebellar nucleus. What could be the purpose? What could be the purpose? So note that there is somatotopy of neurons in the deep nucleus, And what has been found is that when, because of the way there is, because there is somatotropy, and because different Purkinje cell neurons project to different deep cerebellar nucleus, and there is some kind of synchronous activity in the ML direction. ML direction means different groups of Purkinje cell neurons are coordinated simultaneously, leading one to speculate the possibility that there is some kind of multi-muscle coordination that is probably mediated by these neurons. Yet we do not know. I mean, this is only speculation, pure speculation. Okay. So probably this is the kind of things that parallel fiber so because parallel fibers, innervate many of these species, Purkinje cells, it's possible that parallel fibers may be the ones that let the Purkinje cells learn this multi-muscle coordination. This is once again a very intense speculation. We do not know. Okay. Not many students who have joined. I'm not sure if this Manmohan, are you there? You are my only hope. Not many. I think one of the persons who is watching is me. I think there are two others who are watching, but I think they are silent. They just switched it on and maybe are not watching. Anyhow. Okay, there was a question in the discussion forum. It looks like both Marcy fibers and Golgi cells influence the activity of granule cells. What is the difference between them? Both modulate the activity? Well, one has to go back to the anatomy, note how it works. Marcy fibers are so granule cell input is mediated by both Marcy fibers and Golgi cells, yes. But note how the Golgi cells themselves work. I think this is something that is discussed in the recurrent loops within the cerebellum discussion. Let me check where that is. Okay, I think this is a class from a previous class. This is possibly there in the recurrent loops class. So in the recurrent loops within the cerebellum, we discussed this. Note that there is a recurrent loop between the granule cells and the Golgi cells. Okay, it turns out that the Golgi cells themselves are excited by the granule cells, and they inhibit the granule cells again. Is it not? Check my understanding of this. I think this is how it works. So this is a negative feedback loop that is going on. So I would say that the Marcy fiber input is the direct influence on the granule cell output, on the granule cell activity, but the Golgi cell input is kind of a calming influence, is kind of a feedback or a negative feedback influence, reducing the granule cell's response to Marcy fiber input. Okay, that's how I would view this system, because you see, Marcy fiber input on the other hand is almost always excitatory, because this. Not sure if I discussed this. I think I discussed this. The neurotransmitter associated with Marcy fiber is usually glutamate, is it not? Glutamate. So an excitatory neurotransmitter. So it always excites the granule cell, which then excites the Golgi cells, which inhibits. It is GABAergic, and it inhibits the granule cell. This is at least my understanding of the anatomy. So yes, so there is some modulation that is performed by both. So if you are saying, if your question is the granule cell activity modulated by both Golgi cells and Marcy fibers, the answer is yes. But it's not that straightforward, because Golgi cell activity itself is modulated by Marcy fiber activity and granule cell activity. So I would look at this as the Marcy fiber activity as the major input coming to the granule cells, and the Golgi cell activity as a feedback loop, as a break, as a balancing act on the activity of the output of the net output of the granule cell. But note what is happening. Once again, it's quite difficult to make sense of this, because you are having a, I mean, the numbers that we are talking about, billions. You are going to have millions and millions of these granule cells that are, making hundreds of thousands of synapses with, percutations. What could be its purpose? People are still trying to understand. It's very convenient to use this to model the activity of the cerebellum, but there's only so much you can do that. Right. Important. I think we will proceed. If there are no questions, I think. Prajwal, are there any other questions? I'll keep proceeding. If there are any other questions in the forum, you can then post it. I can try to answer that. Alright, so the important role of cerebrocerebellum in modulating the movement. Note how the input is coming to the cerebrocerebellum through the middle cerebellar peduncle. Only one side of the input is shown. How is this input coming in? How is this input coming into the this is the output, middle cerebellar peduncle. How is this input coming in? The input is coming in from the cerebral cortex via pons. Okay, that's what is shown as pontine nuclei on top. So the input is coming from the cerebral cortex to pons, and from pons to the cerebellar cortex. Cerebellar cortex. So from the cerebral cortex, pons, cerebellar cortex. This is the reason why the cerebrocerebellum is also called as the pontocerebellum. I like playing with these words. So in the exam, you should expect some questions of this type. But really, don't hold me responsible in case there are no questions on this. Once again, the usual disclaimer, I actually don't remember what's on the exam, but I usually like these kind of things. These names, terms, recall, this kind of things. So, I would expect a student, a graduate of this course, to remember these things. This pontocerebellum, neocerebellum, cerebrocerebellum, these are all, equivalent. So don't hold me responsible in case this does not show up in the exam. It's still useful thing to know. Okay, so why is this called pontocerebellum? Because, information from the cerebral cortex is received via the pons. Okay. Right. And from that, it reaches the dentate nucleus. Okay. And from there, from the dentate nucleus, it goes to the ventrolateral thalamus, and from the ventrolateral thalamus back to the cerebral cortex. Okay. This is one pathway. And then there's one more pathway. Okay, there's one more pathway in which, so again, very nicely showing that, cerebrocerebellum. So this is the cerebrocerebellum. This is the cerebrocerebellum dentate nucleus. This is the cerebrocerebellum, and that is the dentate nucleus. Okay, it's relatively big nucleus that's shown here. Oh, my pointer is not showing up, is it? Okay, interesting. I'm talking about that. That's the deep cerebellar nucleus. Okay. Which is the dentate nucleus. Relatively large. this is the larger nucleus among the deep cerebellar nuclei. The other one is the next one is the interposed nuclei, which is essentially two nuclei, twins, located next to each other, and then you have the the other nucleus, first stage nucleus. So this output also goes to the ventrolateral thalamus, and from thalamus it goes to the primary motor cortex, shown here. But what also happens is that some of this input actually goes to the red nucleus, and from the red nucleus, yeah, cursor is not visible, Prajwal, I didn't know that, but I have now highlighted. I think now it might be okay. It goes to red nucleus, and from red nucleus there is a projection. So from dentate nucleus, there is a projection to the red nucleus, and from red nucleus there is a projection to thalamus, and from thalamus there is a projection to the primary motor cortex and premotor cortex. So this is the dentato-rubrothalamic, dentato-rubrothalamo-cortical pathway. In this pathway, it is believed that this pathway is probably responsible for things such as mimicry, mimicking of motor acts, things like that. Probably this is responsible for that. Okay. Also, motor imagery, motor mimicking are probably mediated by a loop involving red nucleus. Okay. Not reticular formation, red nucleus. Okay. So again, this picture shows it as if there is a direct projection from cerebral cortex to the dentate nucleus onto the cerebellar cortex. Actually, this is not true. Note the way the projection is given. The cerebral cortex is on the left, and then the projection is to the contralateral side. Note that why is this? Because the left side of the cerebellum is controlling body parts, are coordinating movements that are relevant to the left side of the body, whereas the left cerebral cortex controls the right side of the body. This is the difference between cerebral function and cerebellar function. Okay, something we should remember. That is the reason I am showing here a crossover. So this probably happens at the level of pons, or maybe that's so. But pons is not shown here on the picture, but usually, almost always, the projection from the cerebral cortex reaches the deep cerebellar nucleus and the cerebellar cortex via the pons, not shown in this picture. So once again, let's go back and check where we are with the course. Where are we? Well, we are interested in movements and understanding movements. We know that these movements is probably mediated. These movements are mediated by muscle activity, which is controlled by spinal cord. Motor neurons in the spinal cord, and these spinal cord neurons are controlled further by neurons in the motor cortex, not necessarily through a single synapse. Maybe there may be multiple synapses happening at multiple levels. And this motor cortex is receiving inputs from multiple regions, such as cerebellum, basal ganglia, and association cortex. But before that, there is the light that is shown. That is the decision to move. Who is making this decision? Somewhere in the frontal cortex, this decision to move is made. The more and more you move frontal away from the motor cortex, the more and more muddled the picture becomes. The more and more or the decision making executive. So somewhere in the frontal cortex, the decision to move is made, and this decision is manifested as the actual act. Very critical to understand this nature, because many times our movements define who we are. Movement is probably the only measurable output of the behavioral system. Only measurable output. So far we have seen motor cortex, so far we have seen cerebellum, and we are also seeing, we will also be seeing basal ganglia and cortical association areas in some level. Note that the cerebellum receives inputs from the spinal cord, which is what is shown as the dotted red line here, coming from the movement this here. So that part, good. Trying to see if I can. So So that's what is shown as the highlighted part. That is essentially cerebellum receiving input from the spinal cord as from the spinal cord from various pathways, cuneo-cerebellar pathway, dorsal cerebellar pathway, ventrocerebellar pathway, and so on and so forth. But it also receives inputs from the cerebral cortex. This is the pontocerebellar pathway. This is via pons, and it also projects to the motor cortex. Not shown here is the thalamus, via the thalamus. But of course, there needs to be no mediator between premotor cortex and motor cortex. Direct projection between premotor cortex, as in association areas and executive areas to the motor cortex. So this is how the decision manifestation is made. And the basal ganglia again, receives inputs from various regions, motor cortex, spinal cord, whatever, primarily from motor cortex, but it also projects back to the frontal cortex and motor cortex via the thalamus. Feedback pathways looks shown here. So what we have seen is that, and what would happen if there is lesions of the cerebrospinal? What are the cognitive functions of this? So remember, I already mentioned there is somatotopy in the deep nucleus, and there is multi-muscle coordination in Purkinje cells in the mediolateral direction means what? Many Purkinje cells are synchronously active. That means that Purkinje cells controlling different muscles may be simultaneously activated, leading one to the hypothesis that there is some kind of multi-muscle coordination between and also how surgery to remove tumors in the fourth ventricle sometimes will cause these are benign tumors. When they are removed, sometimes people can do individual leg activity with the individual side of the body, but when one has to do heel to toe tandem gait, that is walking like this, if this is so suppose that is the toe of the leg and this is the heel of the leg. Okay, I am placing one heel over the one toe and then taking the other leg and placing its heel over the next toe and so on and so forth. Right? This is called as a heel to toe tandem gait. In Tamil we call this as adiproduction or whatever. Right? So this is something that these children cannot do, showing that those parallel fibers that cross over to the other side are the only ones that are getting affected because of this, showing the importance of these parallel fibers, that this is actually a relatively small number of parallel fibers. Actually any number of parallel fibers can be considered to be a small number of parallel fibers because we have billions of this. Okay. This is a small number of parallel fibers that cross over to the other side that are probably critical in their role for bilateral coordination. Importantly, what people have studied these lesions, how people have studied lesions is by actually experimentally inducing these lesions in the dentate nucleus by, for example, cooling of the dentate nucleus. So this cooling of the dentate nucleus only delays the movement but actually does not prevent movements. This is amazing. It's truly amazing. It delays the movement but does not prevent the movement. So that means this important output nucleus of the cerebellum, which we think is crucial for coordination, when it is removed, what is happening is that there are delays in initiating the movement, there is poor timing, but actually the movement itself is not stopped. So there's going to be a lack of coordination, a lack of timing coordination, temporal coordination between movements, but not prevention of movements. And that makes sense because from one point of view it makes sense because we would expect a total abolition of movements only if there is a lesion in the cortex, cerebral cortex. Here we are talking about cerebellar lesion. So here probably cerebellum being a modulator of movements and coordinator of movements, there is going to be a compromise in modulation and coordination. Manifestation, this we call this these manifestations as ataxia. Remember, long time ago in one of the earliest classes, we discussed the fundamental aspects of cerebellar lesions, ataxia, astasia, abasia, and so on and so forth. Remember, long time also. One critical manifestation is ataxia, right? So inactivation of the dentate nucleus cause this. People have studied this in many various ways, and people are asking questions like, well, if I what exactly is the purpose of the cerebellum in cognitive function? This is important because because we think sequencing movements and non-movements, sequencing of things probably is mediated by the cerebellum. We like to think like that. So, what probably is a function of this? Something to know. So, timing and timing perception is also probably mediated by. So I mean there is some evidence showing that there are some effects on non-motor functions, but again there's only so much we can do. Okay, solving a pegboard puzzle involves a greater activity in the dentate nucleus and lateral cerebellum than does a simple motor task of moving pegs on the board. So solving a puzzle involves a greater activity than simply making the peg movements. So that means what? That means simply moving the pegs is of some having some effect, but solving the puzzle means it you need to find a solution. You are having a problem that you want to fix, and so on and so forth. Actually causes greater activity in the cerebellum, suggesting that probably the cerebellum is involved in these kind of cognitive tasks. Some very light evidence of this. Okay. So again, important contributions and important hypotheses from Mara and Albos from the old climbing fiber input to Purkinje neurons modifies the response of the neurons to massive fiber inputs and does so for a relatively long period of time. An amount of long-term potentiation or depression that helps you choose what to do, who to respond to, what to respond to. So in that sense, it is believed that the climbing fiber input may be acting as a teaching signal to the parallel fiber, to the Purkinje cells as to how to react to the parallel fiber input. So which parallel fiber inputs to take seriously? I mean, by the way, this is important for all of us. Okay, so we like to think that, who to believe, who not to believe. We like to think that we know. Okay. We'll skip this part. And we have gone through this. I think I just suggested this. Importantly, cerebellum is believed to be responsible for error correction, motor coordination, temporal coordination, so on and so forth. Basal ganglia is responsible for selection or gating of movements. We've gone through this. Recurrent loops. I was thinking of discussing the recurrent loops. So the cerebral cortex, pontine nucleus, lateral cerebellum, and thalamus. And recurrent loop involving the inferior olive, and then recurrent loops involving the Golgi cells. This is what we were discussing in the earlier part of this lecture. Remember what this does? Golgi cells are activated by massive parallel fibers, and they are inhibiting the granule cells. Golgi cells are GABAergic. They are inhibiting the granule cells. So what does it do? Remember, what are you having? You are having. It's important to understand what we are having. We are having billions of these granule cells, and they are all the time making some noise. They are all the time some information is coming from this. Which of these do you take seriously? This recurrent loop involving the Golgi cells is probably responsible for encoding a form of sparse code. What we call in engineering today as sparse code. Critical this, because otherwise, I do not know what to respond to and what not to respond to. After among all those signals, there are only so many. We call these matrices large matrices with a small number of non-zero elements. We call it sparse matrices, right? So that kind of situation. So you are going to have a large number of granule cells, but some of them are going to be modulated with the information from Golgi. So this loop is a very critical loop, giving us information on which Golgi cells to take series, sorry, which granule cells to take series, and so on. So we believe that, again, only a hypothesis. We believe that probably mediating a form of sparse code, implementation of a sparse code. And we've gone through this. So again, vestibular cerebellum and pathways within the spinocerebellar tract, reticular formation, vestibular nuclei, red nucleus and reticular formation. Note the crossovers and non-crossovers that you see. In some cases there is a crossover. Sometimes you have a double crossover. Please note that crossovers in these very important piece. Again, we have gone through this the cerebellar pathways. And the peduncles, again, I love these topics because, this is information, factual information that I expect the students to remember. The corresponding deep nucleus for the cerebrocerebellum is the dentate nucleus, for spinocerebellum is interpost nucleus, and for vestibular cerebellum is either the fastigial nucleus or the vestibular nucleus. And the input peduncle for the cerebrocerebellum is the middle cerebellum. The picture is showing middle cerebellar peduncle. Here is it. It must show at the top, at the pons. Okay, that's okay. Something that Prajwal, note that the output, we have written middle cerebellar peduncle here at the output near the thalamus, on the thalamus side, but not on the pons side. Actually, the correct thing is the input peduncle is the middle cerebellar peduncle, whereas the output peduncle is a superior peduncle. So there we should have superior peduncle. In the place where we have now written middle peduncle, it must be superior, and in the place near the pons we should put middle peduncle. That's okay. Middle cerebellar peduncle is the input peduncle, and the output peduncle is the superior peduncle. Middle peduncle usually is a majorly, input peduncle. For the spinocerebellum, the input is either from the superior or inferior peduncle, and the output is superior peduncle. And for the vestibular cerebellum, the input is the inferior peduncle. The output is also the inferior. Remember that. We have discussed this. I think we will stop here. We will not go into basal ganglia and the details of basal ganglia. We will discuss this in a future class. But remember how the pathways of this is happening. So there is motor cortex that is going to send information to the brainstem and to the spinal cord, and which receives information from thalamus, from the VL area from basal ganglia, and VL area from the cerebellum, and these two are getting inputs from various regions of the motor cortex, and also receiving inputs from the spinal cord. And so again, another picture of the entire story here. It's very critical that you are all able to draw a similar picture and able to map out the diagrams with arrow marks. Critical. I would expect all of you by this time to be able to draw out all these pathways with arrow marks, signifying which is the input, which is the output, what is the direction in which the input is going. By this time, I would expect this kind of information from you. Sadly, we couldn't really have much conversation. I was expecting some people to join. Perhaps people are thinking the meeting is at 4:30 or 4, I do not know. Anyhow, we started on time at around 3:30. It's almost 4:30. There is not much questions. There was also not much questions on the form. I don't know, maybe students are very busy. That is okay. You will get an email from NPTEL with the link of this live session. You can use that to review your understanding. please post your questions on forums. I would love to meet you again 2 weeks from now. Thank you very much for your attention. Have a great weekend ahead and have a great week ahead. Please hang in there and let's continue our preparation for the exam. Remember, 142 students have registered for the exam. I would expect all of you to attend the exam. My best wishes. Thank you very much for your attention.